Hold on, I don't know how to share, record in this.
I've got it set to record already.
Oh, it's you. Yeah, that's right. You're the host. Sorry. Thanks, Dina. So Dina is the project manager for CREST. So you will probably interact with her a lot, and it's great to have her. She's with us half time, and it's great to have her aboard, and she's going to help keep us on track. And you know, also we want to keep track of activities, because we'll need that for our progress report, of course, at the end of the first year, in addition to sort of seeing where how our trajectory looks. Okay, so let me start. So DMAP is basically this. It's this CREST Center for Dynamic, Multiscale, and Multimodal Brain Mapping over the lifespan. You know, this is just sort of a high-level org chart for kind of how we're structured. So there's a sort of a leadership team consisting of myself, Jess Turner, Robin Morris, Vanetta Dotson, and Sergey Pliss. We have an external advisory committee, and we have an internal steering committee. And then Kyle France is working with us on sort of research, education, integration. And then for these types of centers, they require you to have external evaluators where we kind of track our metrics. And then you'll see here at the bottom is where the research piece gets really interesting, I think. We have 3 different sub-projects, including dynamic connectivity, data fusion, and predictive neuroimaging. And I'll go through some examples of what I mean by that in the subsequent slides. There's a lot of different people involved. What we're starting with are the GSU folks who are on this list. These research talks going forward are open, though, so if you have folks that you think would be interested, would like to benefit, just send the we can add them to the email list, and you can just send that to Dina, and she can add them, and they'll just be able to join. The link is open. There's no password. There's a lot of words on these next few slides. I'm not going to go through the details. I will share the slides just so that you can look at it. So that's why I left those on there. But in terms of our high-level goals for the CREST, you know, the first one is to focus on new ways of looking at brain imaging and predominantly methodology, but we also have some very interesting applications that we're targeting as well. We're very focused on integration of education and training. CREST centers are very much focused also on underrepresented groups and trying to build up a STEM pipeline and inspire students and young faculty to pursue STEM career goals. And then thirdly, we're very much interested in interdisciplinary research. And trying to build up structures that can help retain and train students and faculty and integrate with other GSU programs. So, you know, there's a pretty tight integration. You know, our vision is to kind of have this tight coupling between education and research. These are just a few topics here, in this slide where we've got kind of 3 different sub-projects. And, you know, training is kind of at the core of this, really. So, again, as we transition to more in person, I think that will be really helpful, because I'm probably like you. I'm also very tired of everything being virtual. So hopefully we can transition away from that. But even so, we will be, providing videos and providing training opportunities and the like virtually. And then we're really targeting multiple levels. We're ranging from high school, where we're looking at summer opportunities, enrichment programs. Undergraduate, we kind of have divided into sort of an early phase, so something that's sort of not going to be time consuming, not going to take away from individuals who have busy schedules, but maybe just get their foot in the water and see if they're interested. and if we can get them excited about STEM. And then more engaged sort of research opportunities for late phase undergraduates. I just emailed you the summer program that we're going to have information about that. So please do circulate that to your students and to your networks. And then finally, graduate activities. So CREST we're currently reviewing students who have nominated themselves for the CREST graduate fellowships and GRAs. And again, it's not too late. If you have someone, you can let us know fairly soon if possible. But there will be other opportunities throughout the subsequent years as well. And then finally, we're gonna do outreach opportunities. Like we've got one, at the Atlanta Science Festival. We're gonna do hackathons, data competitions, public lectures, and the like. Okay. And so that's that. So now I'm gonna go through just some topics in the sub-projects, which is where things get more interesting from the research side. So I'll just go through each one of the three. So you can kind of see some of the things that we had sort of put in the proposal, at least initially. And I'll start with unimodal brain dynamics. And so here, we're really focused on, dynamics has been, studied extensively in many different fields. But in the functional MRI world, people were not really doing whole brain dynamic connectivity work, until about, I would say, 8 or 9 years ago. It wasn't very prevalent. It's since they've since discovered it in fMRI. So surprise. Essentially, instead of taking two brain regions and looking how they're coupled on average, we essentially want to look at the changes in that coupling over time. And there are lots of ways to do that. And we can do that through something called temporal dynamics, or you can use spatial temporal dynamics, where your nodes or your regions are growing or changing or shifting. The word that we're using to sort of jump on the connectome abuse of the omic notation is the chronectome. Thinking about this sort of broadly speaking. So we can look at dynamics within a particular data set, or we can look at dynamics across the lifespan, right? So there are lots of data sets that we will be able to tap into, and we can look at changes in brain structure. We can look at coupling between brain structure and brain function. We can look at the relationship between dynamic metrics in function and other kinds of changes over time in structure. So there's a whole bunch of interesting things that you can do there. In the dynamic sub-project, we have 3 key goals, and again, these are pretty broad, broadly written and conceived of. But one is to sort of integrate spatial dynamics, because though fMRI has discovered dynamics, most people in fMRI are focused on fixed nodes, and so they basically you'll take regions of interest, and you'll look at dynamic models or dynamical systems models between those regions of interest. But those nodes, because these are functional regions, may change spatially over time. Second is to develop more flexible models. So basically, you can model data with very strong assumptions, and you can model data with very minimal assumptions, and we want to sort of tackle it from both directions. And then thirdly would be, we need some way to summarize the results and provide some intuition so we can understand what this is telling us about the brain. And the focused application in this context is reading, and that's led by Robin Morris. And ultimately, we want to just also, any tools, any techniques that we develop, we want to provide software and tools for people to be able to use those. So just a few samples of the kinds of things I'm talking about. This is just a slide sort of highlighting what I mentioned before. If I have 2 regions, they might be coupled in different ways temporally over time, right? Or maybe their coupling in time is the same, but the regions are shifting or they're moving, right? Or maybe they're changing both in their coupling over time and in the spatial properties of those nodes. And a lot of this work has been done by Armin Araji. He's the one that created this slide, so I want to give him credit for that. And other folks have been working in this space as well, including Robin Morris, I mean. I'm going to get the Robins mixed up as we go forward. We've got two Robins here. And so here's a couple ways that just to kind of give examples of what do we mean by spatial dynamics. And this is, again, some work by Armin. One approach is to think about a hierarchy of brain regions. And so we have a brain network here, say an fMRI brain network called the default mode network. And maybe we subdivide that into smaller components, right? Smaller sets, smaller regions. And the coupling of these regions, the contribution of these regions individually varies over time. And so then that gives you essentially a default mode network that is spatially now varying because it's contributed differently by these different pieces. Or we could look at flow, right? So we could look at essentially the default mode network, and we can allow it to change spatially and over each individual time point. Rather than dividing it up into pieces, it just has voxel-wise variation. And you can look at how it overlaps with other networks temporally or over time. So regions here that are white are contributed to by all three of these networks or states. This one is only contributed to by state 2 over time. In terms of the goal for looking at more flexible models, just again, a couple examples. This is some work by Ashkan Fagiri looking at something called the filter bank approach. And so essentially, instead of looking at, say, a correlation between the time series, fMRI time series from two brain regions, you might expect them to have different contributions at different frequencies. Like maybe there's a high frequency piece that stays strongly coupled the whole time and a low frequency piece that couples and decouples. And so those kinds of models can be captured with this sort of filter bank approach where we essentially get a contribution of different frequencies to a given state. Another approach is to look at decomposing a neuroimaging data set, an fMRI data set into multiple 4-dimensional time series networks. And so if you do that, and this is some work by Behnam Kazimovesh, who's a student working with me, and so this is a sensory motor component, and this is showing the amplitude of one slice in that component, and this is showing, again, it's a 4-dimensional network, and this is showing the variability of that network over time. And you can see some multiple components here within the sensory motor strip that are showing quite a lot of temporal variability, but they get washed out when you look at the amplitude. And then thirdly, looking at metrics, there are lots of ways to summarize all this data. It's very high dimensional. Coming up with intuitive metrics can be challenging. We've been using, simple metrics, things like how much time, if we divide the data into connectivity states, patterns of brain activation, we can ask, well, how long does an individual spend in that state once they enter it? And that's called the dwell time. Or another way to do that is looking at something called fractional occupancy. But we can also look at essentially a subject's trajectory along time and think of it as a state space where we have state 1 versus state 2. And we can characterize this in various ways, like we can compute the total distance that subject traveled over the experiment. And we can do that with an approach called metastates, which Robin Miller worked on. other metrics. If we're thinking about, again, variation of spatial patterns over time, well, you could characterize that with essentially a spatial temporal transition matrix instead of a transition matrix. This is sort of looking at spatial distributions. Or this is some work by Victor Vergara looking at, kind of bringing in a little bit of the dynamical systems theory, but it's really very data driven in this case, but looking at sort of the output from fMRI states as sort of sets of states that have essentially attractor models between them. And we can look at how that varies in, for example, in this case, it was looking at folks who had concussions and how that recovered over time or didn't recover over time. And then finally, the focused application is reading, and this is going to be led by Robin Morris, and we're soon going to be collecting data where we're doing concurrent EEG and fMRI in expert readers, and then are going to be applying transcranial magnetic stimulation and looking at how that impacts the dynamics of the networks. And so this will be a pretty rich data set, multimodal data set, that will be available to be looked at in various ways in the context of the CREST. The second sub-project is multimodal fusion, and here we've got, again, 3 goals. One is develop new models for capturing relationships between multimodal data as throughout the lifespan. The second goal would be develop models that can link data across mismatched dimensionality. So we have, like, for example, fMRI data has a time dimension, a short-term time dimension, whereas structural MRI does not have a short time dimension, but it does change slowly over life, right? And so how do we link those data sets? And thirdly is, again, to increase model flexibility through incorporating things like deep learning approaches. Our focused application, which is led by Vanetta Dodson, is looking at signatures of cognition and mood in the context of multimodal information. And so, if you haven't heard the term fusion or data fusion before, essentially we're talking about combining different kinds of data. Modalities might be like, vision and sound, et cetera. But in the context of neuroimaging data, we might be talking about brain structure and brain function, or we might be talking about EEG and fMRI, or we might be talking about REST data and task data. And there are lots of ways to combine data. You can range from just visual inspection. I've analyzed 2 data sets and I put them side by side and I try to synthesize that in my head. Data integration would be, well, I maybe co-register them to one another if that's possible. So that might be, like PET CT fusion, right? So I put PET on top of the CT data. But the PET data and the CT data those estimates haven't been informed by one another other than to co-register. Asymmetric fusion is something like using maybe an fMRI activated region to seed an EEG source model, right? So then it's one directional. I've used fMRI data as a prior for my EEG model. And then full symmetric fusion would be allowing the data to interact jointly. And this again would be sort of allowing the most shared information, and this would have the least shared information. And we really are interested in leveraging joint information, which potentially in this case, it's just a silly example, but these distributions here, there are 2 groups, and neither of these individual modalities separates the 2 groups. But if you were to combine them together and look in this direction, you can start to see the groups separate, right? And that's really sort of illustrating some of the potential and power of multimodal fusion. And we also want to look at the data, in ways that isn't just univariate. So if we want to look at something, in a multivariate way, all that's really doing is allowing us to look at patterns in the data. You can think about it that way. So here we have these little smiley faces that are sort of coupled in these 2 data sets. So just some examples of subspace models. On the left here, we have multiple network domains in fMRI. So we're treating each of these as essentially a modality. And we've estimated the information between these different network domains. But the model has basically tried to determine whether it can be well modeled by a very simple linear model, or a multiple layers, right? So one or two. So as you move up here, the model becomes more complex, and it requires more parameters to fit. And so one of the things about this is that if we say want to classify, we want to do like age prediction, or we want to predict some difference between groups, we can identify a model here based on this structure, and then it will tell us, well, between the cognitive control and the visual domain, I needed to use a very complex model. So those relationships are more, there's more sort of information, more complexity in that information than between, for example, within the subcortical region here to itself. This is work by Ishan Bada, who's a graduate student. This is another approach called multi-dataset independent subspace analysis. This is work done by Roger Silva. And essentially, the idea is, well, we've got multiple modalities, and we want to allow a model. We wanted to develop a model that can allow us to capture, different types of relationships in that data. So maybe there's a joint source within the diffusion data, and that would be better captured by a model that allows for that kind of a relationship. Or maybe there's a nonlinear source in the data, and we want to be able to capture that. These are just a couple examples of linking data with different dimensionalities. So if I just look at the left here, and don't worry, I know it's a busy slide, but essentially what we're trying to do here is link functional MRI data with structural MRI data while capturing the time series information in fMRI. And so we can do that by estimating, essentially it's an independent component analysis decomposition of the fMRI data, but we essentially link we're sort of allowing the model to jointly estimate the relationship between structure and function, while also decomposing each individual modality into brain networks or covariant networks. Another model here, and this is work done by Lei Wu, is really looking at sort of a connectivity matrix approach, where we have probabilistic tractography from diffusion data, and we're linking it to functional connectivity from fMRI data in a joint model. And she's got some really interesting work where she actually finds these nice tight links between functional networks. This is default mode network from fMRI, and these sort of diffusion tracks that are coming from the probabilistic tractography and the diffusion data. And then up here, we have another model that's sort of trying to link dynamic connectivity with structural information using an attention model in deep learning. And then in terms of signatures of cognition and mood, this is just some work from Vanetta Dotson, where they were looking at relationship between structural information and functional networks, and how those are related to mood and executive functioning. And this is some prior work that she had done, but we are going to be tapping into, as part of these projects, a lot of shared data, as well as the collected data I mentioned. So there are actually, tens of thousands, over 100,000 data sets out there that we can leverage. We've got a lot of it already sort of downloaded and sort of processed here already. And so as part of the CREST, we're going to basically provide individuals, we'll be able to add individuals to those projects so that they can start to look at the relationships in these sort of large data sets and get some more definitive answers. And so there's the ABCD study, which is a longitudinal study of about 11,000 kids. UK Biobank has, their goal is, they've got over 50,000 data sets collected, and the goal is to get to 100,000. Healthy Brain Network is about 10,000 deep phenotyped kids. It's actually ages 5 to 21, I think. And there are many more. And so the third sub-project is really it spans across those other ones as well, but trying to use the imaging data to make predictions. So predictive neuroscience or predictive neuroimaging would probably be more what we're trying to do here. And the goal is to develop multiscale models that it will evolve both within and among individuals, predictive models. We want to avoid sticking with just black box type output, just getting a percent prediction. We want to, we're interested in how the brain works, what parts of the brain are involved, how it's interacting, what it means. And so we really are focused on visualization and understanding the models. And thirdly is more sort of the software tools and to kind of manage the data and develop the data. And so our focused application here is lifespan predictomics. And so with predictive neuroscience, essentially we're trying to, do either classification or regression, predictive regression, using a variety of data sets, complex data sets. And ultimately the goal is to be able to say something about an individual. Or it might be to, to do some sort of evaluation of subgrouping. There's a lot of heterogeneity. And there are, the field, I think, has very much been, it started out with everybody doing group analysis, and then now it's more individual, and then there's subtypes, and sort of you fall anywhere along this spectrum. And there's a lot of information in the data, and oftentimes it's not particularly clear how best to model it, or we're trying to predict something where we don't have a good, understanding of what we're trying to predict. And so that's what this sort of project is about. In terms of multi-scale models, just a few examples. I'll just kind of jump down here. This is again some work by Robin Miller looking at sort of polarity changes in fMRI data as a function of age, and she found that sort of these orbits or these polarity cycles were significantly impacted different in these different age groups. This is another example from MEG data. Where a brain age model, so you can predict chronological age from a normative sort of typical population, and then look at the difference between the brain age prediction and the chronological age as a relationship to predict, for example, behavior. In this case, the flanker effect was predicted by the brain age gap or delta function. Visualization is, as I had mentioned, really important. Alex Fedorov has been working on some deep learning models, and deep learning models have lots of parameters. They're very complex, but he is also working on approaches to take the sort of very compressed encoded information in the deep learning network, and look at how that, and basically decode that back into brain regions and brain networks and interactions between modalities. Similarly, Eloy Ginjar has been working on something called chromatic fusion, which is looking at essentially modeling modalities as colors, and then looking at in that space how we can decode the colors and interpolate essentially between modalities, for example, just as you can interpolate between numbers in this case here. And then the last topic there is essentially software tools. And so for those of you that scan at CABI and are interested in CABI Seed Awards, we're also offering access to these tools as part of those seed grants, where there's essentially a web-based portal. So if you want to work with standardized analysis and kind of have things automatically run, you can do that through a tool called BrainForge. which is connected at CABI already. We're also very interested in, as we're bringing together all these different data sets, harmonization can be challenging. I think the neuroimaging data is just only part of that, because we've got also lots of different assessments, lots of different, ways things were measured. And so we're working on approaches to try to help us do that data harmonization. In terms of lifespan predictomics, we're obviously interested in different age stages. So this is just a little bit of work looking at the relationship between, on the left here, I'm showing you covariating patterns of gray matter. And on the right, I'm showing you functional MRI brain networks, intrinsic networks. And there's actually quite a lot of similarity, remarkable similarity in gray matter covariation brain networks and fMRI co-networks, even though this is presumably just brain structure. You can see these changes that we can evaluate as a function of age, for example. Here's another example looking at predicting brain age from functional MRI data, and you can predict it quite well. So that's all my slides. In the next few weeks, we'll start, we've got some sort of initial talks from the CREST sub-project leaders. Next week, we'll start with a talk on dynamics by Robin and Victor, and then reading, data fusion. February 11th, there's an aging conference where CREST has actually participated in offering support for an award for that. And then February, we'll pick up with cognition and mood, predictive neuroscience, and then prediction of traits across the lifespan. And then starting on March 11th, we'll transition to research talks from students and faculty, and I'll share schedules for that as it evolves. So thanks. Imagine we had a great, picture of the CREST group if we were all together. I should take a screenshot of our, of our video here. And with that, I am going to stop sharing my screen and see if folks kind of had any questions about the CREST generally, or about the slides that I presented, or just wanted to discuss anything in particular. So we'll just kind of open it up now. Yeah, Rogers.
It's a great talk.
Thanks.
I was wondering, could you expand a little bit more on the plans for the educational component? I think you briefly mentioned that there's going to be some kind of teaching involved.
Yeah, so Yep. So there's actually a lot of pieces to that, depending on the level that we're talking about. And so I think when I share the slides, you'll be able to see a little bit more of the details, all those words that I skipped over. But we're going to have training workshops, for high school students, like just sort of like boot camp, introduction boot camp in the summer. We have it's a 4-day session. We're going to, talk about, what is the brain, and how does an MRI scanner work, and the brain and AI, and, things like that. The education at the undergraduate level, we've got research experience for undergraduates, which is that summer program that I had emailed about, which is a mentoring component where students can get exposed to research and learn about brain imaging. Obviously, at the graduate level, we've got training workshops like our fMRI workshop. There are graduate courses. There's already a lot going on at GSU, and we're going to try to sort of highlight that and bring those together by sort of, there are courses taught by by Jess and by Mukesh and myself and many others. Try to organize those and see what else maybe could we contribute. We're going to try to think, not sort of maybe not immediately, but we're going to try to think, is there an opportunity for maybe a concentration, a PhD concentration in neuroimaging? And if so, how would we bundle the courses, and requirements for that? and there's more, but that's kind of a sort of a high level summary of it.
All right. Thank
Sure.
you.
Stunned silence. How's everybody doing? This is the worst part. I wish we had a room with food in it. We could talk and chat, you know. Floor, you could have all the food that you Floor is great at hosting wonderful meetings where people can get together.
Yes, hi Vince. Wow, great talk.
Thank you.
I didn't get distracted, so you keep me very engaging the whole time, you know. I didn't even answer any texts. The level of engagement I have during your presentation.
High praise, definitely in the day.
Yeah, no,
Hard
it
to
was
keep me
a
from getting a
very,
text. So yeah.
very engaging, a great summary of the work, you know, it's been done.
Mhm.
And yeah,
Thank you.
I'm looking forward to keep working in the predictive models.
Yeah.
Also,
Yeah.
you know, if any contribution I can do during, you know, preparing courses for undergraduate students, or, you know, giving talks, you know, I'm bilingual, as you know. I'm very
Yeah.
good talking to people with different backgrounds, you know, minorities and Hispanic and all of that, you know, any contributions I can do, you know.
Yeah.
I
Absolutely.
can make.
Absolutely. That's a
I am
great.
here from
Yeah.
New Mexico.
Yeah.
And you know, flexible to travel as well. But I just want to know, it's very interesting, you know, and I'm glad. Thank you for inviting me to the meeting, and I'm looking forward to learn what is being done and what I can collaborate
Yeah.
or
Yeah,
contribute.
and for those that don't know, Flora also has gone to Peru to give some lectures there, right? You had done some.
Mhm. Yeah,
So
and I'm looking forward, you know, stuck with the results for the alcohol data, but I'm looking forward to get results, get the paper, and prepare a talk in Spanish, you know, like your brain can, you know, differentiate people who drink, those who don't drink, you know, borrachos and no borrachos in Spanish, you know, will be a good way to capture people interest in neuroscience, you know,
Yeah.
presenting in a friendly way, you know. So.
Absolutely. And we'll have you give a talk here, though maybe not in Spanish.
Yeah, I can do both English, but yeah.
I would probably get every maybe 10th word, or if that. So yeah.
Yeah, looking forward down the road to be able to, you know, put a talk in and share what I'm doing. Thank you.
Great.
And
And yeah,
I love eating. Yeah, Vince was right. I love eating and setting up wonderful, delicious meals.
it's true. It's true. Yeah. Some of the best meetings we had were at Flora's place. So yeah.
Oh, great. Thank you. That you made my day. Thank you.
For those that don't know, I was in Albuquerque, and Flora is still in Albuquerque. So we used to be kind of co-located. So yeah. And I guess just to echo what Flora had mentioned, the reverse of that, which is if anybody has any ideas, suggestions, thoughts, input, share it with, you know, please feel free to share it with us. You know, as the other CREST leaders know, I don't know what the heck I'm doing most of the time, and I'm trying to do my best, and I'm trying to respond and adapt to and learn as I go along, and see if we can sort of maximize the opportunity here. So I'm certainly, I certainly think we're stronger if we, you know, provide input. Everyone provides input. So does anyone else on the you know, in the CREST group want to say anything about, maybe, I don't know, Kyle, if you wanted to say anything about the educational piece at all? And I guess I'll put you on the spot, but
Sure. Sorry, I am en route to my home in Decatur, so I'm just on audio. Sorry, but yeah, I missed the first part with conflicts this afternoon. I imagine that you talked a little bit on all the different education programs, and I just want to say that this team has been so attentive to the opportunities to share this knowledge and bring new people into this field, that it's really impressive. It's something wonderful to be a part of. And I know that as the group gets underway, you'll tap into the rest of the resources that the other faculty and their trainees bring to this field, and really enable us to reach students at the K through 12 level, not just through the high school program that you mentioned, but also we have a couple of events coming up in the Atlanta Science Festival already, and then some other links to the community through mentoring programs that other faculty have raised. And then at the undergraduate level, starting with this summer research program, but we're also planning some courses to introduce and would need ideas for what kinds of curricula would fit there and how to reach students on all our campuses of Georgia State University, not just the downtown campus. And then you all are already just so impressive with the graduate and postdoc training, but the funding that is provided with the CREST really amplifies the opportunities to continue to train at that level and accentuate the postdoc transitions into faculty positions or other roles that are going to be critical to getting this information out there and really advancing knowledge in these fields. And the diversity, equity, and inclusion arm of all of this has really shown through already. And I think we're establishing procedures that will minimize bias in recruitment and retention of individuals in this field. So kudos to all. It's been a pleasure to be involved. Thank you.
Yeah. Yeah, and I also I'm really happy to see that we have folks on here from a variety of departments. We're really trying to be as broad as we can at GSU, and because knowing that there are lots of people working in different areas, in the business school, neuroscience, math, computer science, psychology, et cetera. So it's really great to see that, and I'm hoping again that we can really sort of bring together folks across departments to contribute in this space. So
if I could put a shout out for the Aging Research Conference that you mentioned, February 11th. Yeah. I'll be sending around flyers and everything hopefully next week about it, but we will need a couple of people to volunteer to be judges for the neuroimaging prize. We've got the Trends or DMAP is supporting the best poster for neuroimaging research and aging, and I could certainly use a couple of 2 or 3 people who could volunteer to be there that Friday and help judge.
Yeah. And we could send out an email to the CREST email list as well.
Okay.
So just in case you aren't sure yet and you want to take time to think about it.
Yeah, well, there'll be more details next week.
Yeah.
Sure.
Right. Yes, if you know what's a time requirement, then maybe the people can more clear know whether they can.
Oh, yeah,
do they have to show up in person? Is it virtual? Can they do it? Is it a 10-hour thing or a 2-hour thing or a 1-hour thing?
We'll have to be there in person at least but it's Friday it's Friday, February 11th, and the posters are from 1:30 to 2:30 in the afternoon. So we're actually meeting the program committee is meeting next week to sort of work out the judging details of exactly how we're going to get this all judged in an hour, but it shouldn't be much more than that in terms of a time commitment.
Excellent. And I see quite a few people on who I will probably ask to prepare some talks for the subsequent research talks, so that'll be fun to kind of see what everybody's up to. So
Yes, just a comment. I'm surprised that Victor doesn't have any questions.
he does. He just hasn't shared them with us yet. So
I just hope I don't have many questions next Friday.
Yeah, Victor's presenting with Robin next Friday. So for those of you that are just to kind of update you on the status of the graduate research assistantship process, we're working on doing some evaluations right now. So we will hopefully have some additional information to share in the next week or two, I would think. So depending on if we can get our ducks in a row.
if I don't mind me butting in again, I just got a notification of a special call for a research topic in Frontiers on methods and application in aging psychiatry. So I'll forward that out to everybody.
All right.
If they want to participate in it.
Yeah. Oh, and yeah, like I said earlier too, if you have other people that you want to join these meetings, or you think they would like to join, just send the emails in. So we can certainly add them. And if we add you and you want to get off, we'll let you get off. So don't worry, you're not locked in forever. Any other things that folks want to share?
So I'm guessing we can invite students to events?
Yes. Yeah, absolutely.
That is relate my question is, other presentations we're going to give is targeting to other research or the seniors or professors, or targeting the undergraduate students, that's a different level of understanding.
Yeah. I mean, it's always hard, but I would try to keep it, a little bit general as if you can, so that we can sort of target as broad an audience as possible. But we're gonna, in some cases, we'll get into some details that are more specialized, and I think that's fine too. So.
Okay, more generalized. Good.
Yeah. All of the above, I guess. And I can certainly share details about the talk ahead of time if that's helpful, right? We can email it out so people will know, oh, this looks interesting to me, or I want to learn more about this, or it's in my area, etc. So, and anybody else want to share anything? Andrea, are you gonna do any? Andrea, are you gonna do any neuroimaging with us? Andre, did I say that right? Andre Shilnikov. Hello.
Hey guys.
Hey.
Hey.
We're gonna try to pull you into neuroimaging. So
I don't do neuroimaging.
I'm gonna change that. I think the stuff you do, there's some potential connections we can find. So.
All right.
Yeah. Pair you up with a student, maybe. So.
Maybe yes. I'm in DC now, visiting my daughter.
Ah, okay, great. Everybody's all over the place here. So.
Exactly. Let's talk. Yeah, and finally we have to all meet together one day. Enough of offline meetings, right? Enough.
Yes, totally agree. Okay, great.
I mean, again, you raised a very good point, how to connect imaging and dynamics, and I'm really interested in that trend. No doubt about that.
Yeah. Yeah, I think if we can bring in some students who are not afraid to kind of cross disciplines a little bit, then we can do some really neat stuff. So.
Yeah, I mean, one of the students is David, that we co-advise with Robin, right? And he's a perfect match for that task. Absolutely.
Okay, great. Any other input or thoughts? If not, I think that's all I had for today. Thanks again for joining. And I'll send a reminder next week, or Dina will send a reminder next week about the dynamic session that we'll have. So.
Perfect. Great. Thank you.
Thanks. Okay. All right. Thanks, everyone. We'll adjourn for now. And then for those of you that are part of the CREST leadership, please stay on. We'll just continue in here. So. Got to turn off that sound. Beep, beep, beep.
Yeah, sorry, I was thinking the same. I need to figure that out.
Yeah, there's a way to do that. I think I always forget, when I'm in the middle of a meeting, but I see it at some other point. So. All right. And, all right, Sarah, are you there? she may not be at her desk. So, well, that's fine. We'll just go ahead with the discussion anyway. Sarah's a postdoc in my group, so she's not one of the graduate students. I guess I just wanted to touch base about the graduate student evaluation process that we.
Hey, Vince, real quick, do you want to keep recording at this point? I don't know.
Yeah, Dina, maybe let's stop the recording. Yeah, we can always